Enzymes are characterized by two properties. One, they are extremely specific. That means a given enzyme will only catalyze one type of chemical reaction. Two, they are very effective since they can increase the rates of reactions by 1,000,000,000 times to millions of billion times, depending on the type of reaction, the site where it occurs, and the time the product is needed. To make an analogy, suppose that there is a baby boy born in the year 2010. This baby will grow up, becoming a child, a teen, a young man. He will attend schools and colleges, he will work and have a career, and he will eventually retire. After a while, he will be celebrating his 100-year birthday. So the rate of reaction to change the baby to an old man is about 100 years. If there was a magic catalyst equivalent to a specific enzyme, that would speed up the lifetime of this person. The time of 100 years will be reduced to only 3 seconds. How do we explain this rate change? Here is a graph representing the energy versus the path of reaction. The reaction is reactant A yields product B. Case without catalyst. The reaction shown here is an exothermic process since the enthalpy change is negative. In the other hand, the reactant A must absorb a certain amount of energy called EA or activation energy and changes to an excited state A star, also called transition state A star. Then this transition state yields the product B. In the presence of catalyst, the catalyst lowers the activation energy. The curve is changed from the red color to the teal blue color. The excited state A is reached just after the reactant A absorbs the amount of energy shown in the red arrow. That has no effect in the enthalpy change, but speeds up the reaction. As we stated before, that may go easily at least by 1 billion times the initial rate. So the activation energy in a catalyzed reaction is reduced. Two factors will be determinant for the enzymatic activity: temperature and pH. The temperature of a human body is 37 degrees Celsius, and so is the temperature inside cells and different organs where reactions take place. The graph shows an optimum activity at 37 degrees Celsius. This activity decreases below or above that temperature of 37 degrees Celsius. The blood and fluid in the body has a pH 7.4. The pH of most sites in the body equals 7.4. The graph shown here tells us that the enzymatic activity is optimum when the reaction occurs at pH equals 7.4. In the contrary, in the stomach, for example, where the pH is very acidic because of the presence of hydrochloric acid, the optimum activity should be around pH 1.5. The purpose of this lab experiment is, first, to observe different reactions. Second, to use different techniques in order to detect the enzymatic activity. The lab itself is divided in 3 parts, Part A, B, and C. In Part A, you will be doing the hydrolysis using 3 enzymes, trypsin, pepsin, and papain. Part B, hydrolysis of fat molecules found in milk. And Part C, hydrolysis of urea. Let's see in detail those different reactions. Part A. Papain is the specific enzyme used to break down the peptide bond of the amino acid cysteine. That means the papain will catalyze the hydrolysis of cysteine peptide bond. In other words, the primary structure is broken at the cysteine site. For example, we have here a tetrapeptide, and you see the cysteine in blue color. When you use the papain to assist the hydrolysis, the bond will be broken at the cysteine peptide bond. The trypsin catalyzes the hydrolysis of lysine and arginine peptide bonds. In this example, we see those 2 amino acids, lysine and arginine. When the hydrolysis occurs, assisted by the trypsin, the peptide bond at the lysine site will be broken, and the peptide bond at the arginine site will be broken as well. The last enzyme that we will be using in Part A is a pepsin. Pepsin catalyzes the hydrolysis of 3 amino acids, phenylalanine, tryptophan, and tyrosine. In this example, we see those 3 amino acids, the phenylalanine, the tryptophan peptide bond will be broken as well as the tyrosine peptide bond. On Part 2, you will be performing the hydrolysis of fat molecules found in milk. The enzyme that will be used here is the pancreatin. The pancreatin catalyzes the hydrolysis of the ester linkages in fat molecules. Below is the reaction between the triglycerides, fat, water, assisted by pancreatin. This is a hydrolysis that will yield fatty acids and glycerol. You will test the presence of fatty acids in this reaction. The third part is the hydrolysis of urea using the urease. Here below is the reaction between the urea and water assisted by the urease. Normally, if the reaction occurs, the products are ammonia and carbon dioxide. So in this experiment, you'll be testing the presence of ammonia by using an acid-base reaction. Before we start the lab experiment, these are the precautions that must be taken. Goggles must always be worn until you leave the lab room. Be careful when handling chemicals. Some of them can irritate your skin. Wash with a lot of water if something spills on you. Nothing goes down the sink. Don't forget to use the waste container. And finally, clean up your workstation before you leave. Now put your goggles on and let's get started.